Hello friends, welcome to my YouTube channel Medicinus Tips. Today we are going to look about glycolysis. We will learn about the steps of glycolysis and enzymes involved in different steps of glycolysis. We will also look the net ATP balance sheet of glycolysis in aerobic and anaerobic. We will cover the individual steps of glycolysis in detail. So, glycolysis is the process of formation of pyruvate or lactate from glucose. It is the first reaction in respiration. It occurs in almost all living cells. It was the first metabolic pathway to be sequenced. It was first described in 1930. It is also called EMP pathway, that is, it was first described by Gustav Emden, Otto Meyerhof and J. Parnas. It occurs in cytosol. The enzymes of glycolysis are present in cytosol. The glycolysis is a central metabolic pathway. Many of its intermediates provides link for other metabolic reactions. The intermediates of glycolysis are useful for the synthesis of amino acids and fats. The glycolysis, it occurs either in presence or absence of oxygen, that is, it can be aerobic or anaerobic. Lactate is the end product of anaerobic glycolysis, while in aerobic glycolysis, pyruvate is the end product, which enters TCA cycle and gets oxidized to carbon dioxide and water. The glycolysis is an important process for ATP formation in tissues lacking mitochondria like RBCs, cornea and lens. As we know, glycolysis occurs inside the cell, that is in cytosol. Now we will look how the glucose gets transported from blood into cells. It is a concentration driven process. It occurs by facilitated diffusion of glucose from high to low concentration, that is from blood to cells. Usually the glucose level is high in extracellular fluid, but it is low in gluconeogenetic tissues like liver and kidney during certain conditions like fasting. It is done by glucose transporters. There are usually 5 glucose transporters described: GLUT1, GLUT2, GLUT3, GLUT4 and GLUT5. GLUT1 is present in most of the cells. GLUT4 is an insulin dependent which is present in skeletal muscles and adipocytes. GLUT2 is insulin independent which is principally present in liver. Phases of glycolysis. There are 3 phases for glycolysis. First phase is energy investment phase, also called priming phase or preparatory phase. Second is splitting phase and third is energy generation phase or payoff phase. During energy investment phase, ATP is used, while in energy generation phase, ATP is synthesized. This is an overview about glycolysis. We will look into each steps of glycolysis individually in detail and the enzymes involved in that steps. Glycolysis is a sequence of 10 enzyme catalyzed reactions that converts glucose into pyruvate with the simultaneous production of ATP. In glycolysis, 1 mole of glucose is partially oxidized to 2 moles of pyruvate. The anaerobic glycolysis reaction is glucose plus 2 ADP plus 2 inorganic phosphate gives 2 lactose plus 2 ATP. The reversal of glycolysis with alternate arrangements at irreversible steps lead to gluconeogenesis. First is energy investment or preparatory or priming phase. In preparatory phase, there are 3 reactions. First is phosphorylation of glucose, second is isomerization of glucose 6-phosphate and third is phosphorylation of fructose 6-phosphate. Step 1, which is mediated by hexokinase or glucokinase. The glucose obtained from diet through the intestinal hydrolysis of lactose or sucrose or glycogen is brought into the hexose phosphate pool by the enzyme hexokinase, which converts glucose into glucose 6-phosphate. The reaction is phosphorylation. The glucose is phosphorylated by the enzyme hexokinase or glucokinase with the utilization of ATP to form glucose 6-phosphate. The glucose 6-phosphate formed is impermeable to cell membrane, so it gets trapped inside the cell. The reaction is an irreversible reaction in which magnesium ions acts as the cofactor. The glucose 6-phosphate formed will enter into different metabolic pathways. The fates of glucose 6-phosphates are glycolysis, gluconeogenesis, glycogenolysis, glycogenesis, HMP shunt pathway. As we said, the step 1 of glycolysis may be mediated by hexokinase and glucokinase. Now we will look the differences between hexokinase and glucokinase. The occurrence. Hexokinase is present in all tissues while glucokinase is present in liver and pancreatic beta cells. The Km value. The Km value of hexokinase is low while that of glucokinase is high. So the affinity of hexokinase will be high and the glucokinase will be low. The specificity of hexokinase is that it acts on glucose, fructose and mannose, while glucokinase acts only on glucose. Hexokinase cannot be induced while glucokinase is induced by insulin. The actions are hexokinase acts even when blood sugar is low while glucokinase acts only when blood sugar is high. Step 2 is isomerization. Glucose 6-phosphate is isomerized to form fructose 6-phosphate in the presence of enzyme phosphohexose isomerase. The reaction is reversible. It's an example for aldose-ketose isomerism. Step 3 is phosphorylation of fructose 6-phosphate. Fructose 6-phosphate is again phosphorylated to form fructose 1,6-bisphosphate in the presence of enzyme phosphofructokinase with the utilization of an ATP. It's an irreversible reaction. It is the rate-limiting enzyme of glycolysis. The phosphofructokinase is an allosteric, inducible and regulatory enzyme. It is one of the key enzymes of glycolysis. The phosphorylation of fructose 6-phosphate is the committed step of glycolysis. That is, once fructose 1,6-bisphosphate is formed, it should undergo glycolysis. It cannot enter other metabolic pathways. It is also called bottleneck of glycolysis. Phase 1 of glycolysis is over. Now we will look the phase 2, that is splitting phase. Step 4 is splitting of hexose, that is fructose 1,6-bisphosphate is splitted into two 3-carbon compounds, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. It is catalyzed by the enzyme aldolase. The reaction is reversible. The aldolase is an example for lyase type of enzyme. Step 5 is isomerization. Dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate in the presence of enzyme triose phosphate isomerase. It is a reversible reaction. Now after the 5 steps, glucose is oxidized to form 2 molecules of glyceraldehyde 3-phosphate. Usually the step 5 of glycolysis is included under splitting phase. The last phase of glycolysis, that is energy generation phase or payoff phase. Step 6 is dehydrogenation and phosphorylation. 2 molecules of glyceraldehyde 3-phosphate undergoes dehydrogenation and simultaneous phosphorylation to form 2 molecules of a high energy compound 1,3-bisphosphoglycerate. The enzyme involved is glyceraldehyde 3-phosphate dehydrogenase. The reaction is reversible. In this reaction, an organic phosphate is added to the substrate. 1 molecule of NADH is produced, which is produced by the reduction of NAD+. The NADH produced will enter the mitochondria via malate aspartate shuttle in aerobic glycolysis. In anaerobic glycolysis, NADH is utilized by lactate dehydrogenase in which NAD plus is produced. Step 7 is the substrate level phosphorylation. The energy of 1,3-bisphosphoglycerate is trapped to synthesize one molecule of ATP by the enzyme bisphosphoglycerate kinase. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. The reaction is reversible. The only kinase in glycolysis which is reversible is bisphosphoglycerate kinase. It is an example of substrate level phosphorylation, that is, production of ATP by the direct addition of inorganic phosphate to ADP without involvement of electron transport system. Step 8 involves isomerization reaction. 3-phosphoglycerate isomerases to form 2-phosphoglycerate under the influence of enzyme phosphoglyceromutase. The reaction is reversible. Ninth step is dehydration reaction. 3-phosphoglycerate undergoes dehydration to form phosphoenol pyruvate. The enzyme involved is enolase. The reaction is reversible. One molecule of water is formed in the reaction. Enolase is dependent on magnesium and manganese ions. Fluoride ions inhibits enolase by removing magnesium. Final step of glycolysis is dephosphorylation reaction. Phosphoenol pyruvate is converted into pyruvate by the enzyme pyruvate kinase. It is an irreversible reaction. It is the second substrate level phosphorylation of glycolysis in which two molecules of ATP are formed. There are three irreversible steps in glycolysis. They are glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate, and phosphoenol pyruvate to pyruvate. Looking at the energy yield in glycolysis, there is a net gain of 2 ATPs. That is, there is consumption of 2 ATPs and gain of 4 ATPs. So there is net gain of 2 ATPs. ATP consumption occur in first and third step, while ATP gain occur in 7th and 10th step of glycolysis. That's it. We have covered the steps of glycolysis and enzymes involved in the different steps of glycolysis. I hope that you understood about the glycolysis. Please like this video and subscribe to my channel Medicinus Tips. Once again, thank you for watching the video.